Hello everyone! Welcome to MaxWell Webinar. My name is Obien and I will be your host for today. I would first like to introduce to you our webinar tool. As you can see here on the right side, you can type in your chat for questions and they will be private. So only the administrators and us would be able to see your questions. Also, if you have any comments about the webinar, feel free to write them there. I see that now many people are logging in. Thank you very much for attending our webinar today. All right, so this is our agenda for today's webinar. So first, I will introduce to you our company, MaxWell Biosystems, and our core technology, high-density microelectrode arrays, and how it is applied for retina research. Next, we will have the main part of the webinar, which is a presentation from Dr. Szilard Sajgo. He will be talking about assessing retinal function in disease and health at single-cell resolution. So this will be mostly talking about the use of high-density microelectrode arrays for retina research. After that, we will have a 10-minute question and answer portion wherein we will choose some of the questions that will be here in the chat. For those questions that are not answered during this webinar, don't worry, still feel free to write them there. We will be able to answer them by email as soon as possible. Okay, let's start. Okay, so we are MaxWell Biosystems, and it is our mission to provide electrophysiology systems and solutions to advance basic research and for drug discovery. Our core expertise includes microelectrode array platforms, which is both on the technology side, that is the hardware and software development for cell functional assays, and also on the application side. So we have expert scientists that are able to design experiments, protocols, and help you in your research. Right, okay. So what do we have? What is our technology? So it is our aim to be able to analyze every cell in your sample. On one hand, the gold standard for functional analysis of cells is electrophysiology, patch clamp, wherein you have a glass micropipette that is placed close to the neuron to be able to detect its activity. On the other side, to be able to analyze these cells, you also would like to see its morphology and how they are growing and connecting with each other. So our technology is in between these two technologies. We have high-density microelectrode arrays, and our products are called Max 1 and Max 2. So here, I'm showing to you a video wherein an action potential is starting at the axonal initial segment and propagating along the axons. So this is electrophysiology, but you are doing functional imaging, so you're able to also extract the morphology of the cell. And here in the inset, you can see multiple action potentials from different cells. So what do we measure? As I mentioned, we measure action potentials. For patch clamp, wherein you're looking at what's inside of the cell, that looks like a positive spike because there is an influx of sodium into the neuron during an action potential. With extracellular recording, which is what we do, we have electrodes outside such that these spikes are negative as shown here below. For high-density microelectrode arrays, the main advantage is that we have many of these electrodes that are able to detect signals not only from one spot of the neuron, but from many different areas, many compartments of the same neuron. In this case, we can see signals coming from the axonal area, the soma, and also the dendritic area. And in this case, we are able to functionally image each of the cell and to be able to find out if these so every cell in the sample are connected with each other. So what are the key advantages of high-density microelectrode arrays as compared to low-density microelectrode arrays? So first of all, here on the left side, sometimes there are cells that are not detected by any electrode because it's in between them. For high-resolution MEAs, we can see all the cells because we have electrodes that can be very close to every neuron that is in the sample. Additionally, here you can see that there are 3 different cells close to one electrode. You will not be able to detect whether the signal that you're getting is from one cell or from 3 cells. However, with high resolution, you can identify the activity of each cell because we have smaller electrodes that are closer to these neurons. That means you can identify if the spikes are coming from the green, the red, or the blue cell. Also, you have electrodes everywhere, so you're always at the right spot, and you have access to the small signals of the cells, such as axonal action potentials propagating along the axons. So these are our products, Max 1 and Max 2. Max 1 is our single well high density microelectrode array platform. We have here the recording unit, which is the data acquisition hardware, and this is the consumable, which is the chip, Max 1 chip, wherein this is where you put your sample to be able to record from them. Here on the right side, we have Max 2, which is our multi-well version of Max 1. We have the mainframe, which is the data acquisition hardware, but also it has a built-in incubator, so the bottom and top plate is heated, and there's also an input for your gas so that you can control the environment during your recording. And then we have the consumable, the well plates, which come in 6 and 24 modes. So focusing more on our high-density microelectrode array, this is showing Max 1, but it's the same microelectrode array that is in Max 2. So every well we have this chip, wherein in the 2 by 4 millimeter square area we have 26,400 electrodes, and the distance between these two electrodes center to center is 17.5 micrometers. That means that for one cell, there can be many electrodes that are measuring signals from that single cell. And what's more important is that we have low noise readouts, 2.4 microvolt RMS. This means that even the smallest signals of the cells can be detected by these electrodes. Additionally, any of these 26,400 electrodes can be used for electrical stimulation. And because the electrodes are small, you can really selectively stimulate single cells in your sample. Central to our products, both Max 1 and Max 2, is the software, MaxLab Live, and we have an all-in-one solution for that. So MaxLab Live is used to visualize and acquire high-quality data, organize our experiments, and run their recordings, and also analyze and export the datasets. So these are some of the screenshots of our software. This is showing the traces view where you can appreciate the action potentials from many different electrodes. And these are other visualizations such as electrodes view and raster view. In the electrodes view, you can see a grid of 26,000 electrodes, and the red spots correspond to the active spots during a burst. Here on the right side, we have the raster plot. Each line corresponds to an electrode, and each dot corresponds to an action potential. So here, the vertical bars correspond to all of these thousands of neurons that are active at the same time during this period. So what are the applications of our systems? 2D cultures, such as those using human iPSC-derived neurons and primary neurons. Tissues, such as brain slices, brain organoids, and neuronal spheroids. And retina. So this is our main focus for today, the application of our high-density microelectrode arrays for retinal applications. So what is the advantage of using our system for retina studies? First of all, we have access to large retina areas. So if this is a retina that is cut in this flower fashion, this is the size of our electrode array. So that's how much of the retina you can record from. Also, we can detect every cell because of the resolution of the electrodes. Here you can see that these neurons is a specific cell type that has been labeled, and they can be very nicely detected because our electrodes are at the same density or even higher density as these neurons. Additionally to our MaxOne system, which is used for retinal experiments, we have the tissue holder to be able to hold the retina flat onto the microelectrode array and to have a stable setup all throughout your experiment. So, how do we do experiments? We use MaxLab Live neuronal assays. I will introduce to you each one. We have four of them that I will introduce today. So, the first one is activity scan. Here we ask, are the neurons active? From here, we can extract whole sample activity map label free. So, here on the right side, you can see on top a microscopy image of a cell culture, and at the bottom, the functional image from either Max 1 or Max 2 that can be extracted from the same culture. So here, not only do you see the cells, the location of the cells, but also how active they are. So the red spots correspond to the ones that are more active, and the light green spots correspond to those that are less active. So here on the bottom left side, we see how a functional image of a retina patch looks like. From there, you can identify where your retinal ganglion cells are and just keep on recording from them. And then use the next assay, which is our network assay. Here, normally we ask, is the network functional? But for retina, we can ask, are the retinal ganglion cells responding to light stimulation? So you can just record from many of these retinal ganglion cells at the same time as you are producing light stimulation. So here on the right side, we see a response of a retinal ganglion cell to light. So this is light on and light off. So this is an on-off cell where it is active at the first part of the on light, and it has this activity during the off state of light. So these can be used to extract a retinal ganglion cell mosaics. And here at the bottom right, you can see the spiking, kind of the bursting of each of the retinal ganglion cells for different directions of light, like going this direction, this direction, this direction, and so on. And this is a video of such an experiment wherein light is being moved from many different directions, and you will see how the retinal ganglion cells react to this light stimulation. So here is moving like this, the light, the other one is moving like this, and so on. Yep. Okay. Another one that is very special to our system is the axon tracking assay. Here we ask, are the axons functional? And we can use this to extract axonal arbors and axonal action potential propagation velocity. Here on the bottom left side, you can see what we call kind of an electrical image of a single cell, in this case, a retinal ganglion cell. And we can also follow the action potential propagation along the axon here. And you can use this to extract the morphology of many cells in your sample as shown here on the right side. So here you can see that all of the axons are going towards the same direction, which is towards the optic nerve. And lastly, we have electrical stimulation as a feature of our system. Here we ask, can we control the activity of neurons? And here, because we have small electrodes, we can really selectively stimulate single retinal ganglion cells. In this case, we have this electrical image from a single cell, and then we target the largest area here for stimulation, and we can appreciate here that the same signals can be detected from the same cell that is stimulated with light and the one that is stimulated with electrical stimulation. So you can choose which neuron you would like to stimulate and stimulate that one cell only at a time. So these are the different features of our system that are very useful for retinal applications. We can access every cell in the sample, we have flexible stimulation, and we also have available data analysis tools for retinal experiments. Okay, so that's it for the introduction. Next, I will introduce to you our application scientist, who is also an expert in retinal experiments, applications, and research, Dr. Szilard Sajgo. So, Szilard, I move to
Hi,
you.
Mary. Thank you for the introduction. So right now, I will just talk about assessing retinal function in health and disease at single cell resolution, and in general, the readouts that you can have from the retina. And first of all, why study the retina, and what is the retina? So the retina, the human retina, is this 300 micrometer thick tissue at the back of the eye that basically translates images into electrical neural impulses and sends them to the brain for visual processing. And one can ask the question, what drives vision research? First of all, it's the social economical aspect of it. This is a huge because some sort of visual impairment affects approximately 285 million people worldwide. As it is evident in these pictures, diseases like age-related macular degeneration, diabetic retinopathy, or glaucoma significantly reduces the quality of life of patients. And of course, the other aspects of vision research is driven by curiosity. As early as Descartes, scientists wanted to understand how visual perception works, and this itself can lead to better understanding on how our brain works. And because the optic vesicle during development is part of the central nervous system. Uh, this can really help us to better understand these processes. Some other advantages of performing experiments in the retina include, despite being a tissue at the back of the eye, we know a lot of facts about it. We know that there are cell types, that there are roughly 60 cell types. These are well characterized morphologically and physiologically. And of course, we know that how these cell types respond to light or light stimulation. Uh, Also, if you take the retina and perform an explant, an ex vivo preparation, it will survive from several hours to even days, depending on how successful your preparation is. Now, retina and MEA technology was studied since the late 1980s, when Marcus Meister and Jerome Pinet performed the very first salamander retina recordings on MEAs. Retina's responses to light can be very easily measured, and they come in the form of micro ERGs and spikes. Now let's discuss a bit in more detail of what these actually are. So the micro ERGs is composed of a sum of electrical potential changes of different retinal cell types. And it is composed in general of three major components. You have the A wave, which is generated predominantly by rod photoreceptors. You have the B wave generated by on bipolar cells, and then you have a slow decaying C wave attributed to retinal pigment epithelial cells. ERG can be used, and it is used to determine which neuronal elements of the retina are the cause of a specific pathological condition, and these recordings are also used in the clinic. Um, and also, with invent of modern pharmacology, we now can selectively block different signaling pathways in the retina and analyze how these affect the micro ERG waveforms. Now, for instance, we have specific B wave blockers in the form of LAP4, which is an on bipolar cell blocker, or for instance, the NQX, which is an selective off bipolar cell blocker. The second main readout from the retina are, of course, the action potentials of retinal ganglion cells and polyaxonal amacrine cells. And also, via these response properties, we can assess indirectly other amacrine cell types like starburst amacrine cells. Now, by analyzing the spiking activity of retinal ganglion cells, we can determine what functional retinal ganglion cells we are actually looking at. For example, are we looking at a directive selective cell or a non-directive selective cell? We can determine if the RGC, what kind of temporal light response properties it has. We can also define its axonal conductance speed by axonal tracing and so on. Now, one can ask, in what context can these readouts be useful for scientists, right? So in the end, they have to be useful in some way. Well, these readouts can be performed in the aspects of drug screening. You can perform drug screenings. You can analyze disease phenotypes using the retina. You can study visual restoration success rates with the retina. You can investigate different aspects of visual development. You can study visual processing in the retina, and you can also perform optogenetic or electrical stimulation experiments. Now, I'll try to give you some example of these applications and how they were applied in peer-reviewed publications. For instance, the beautiful work of Berry et al. 2009, and in this study, they are using the RD1 mutated mouse line where the photoreceptors are degenerated. And what they do is they take a green opsin, medium wavelength responding opsin, they package it into an AAV, they transfect it into retinal ganglion cells of these animals. And as you can see in the raster plots, once these opsins are transfected into retinal ganglion cells, uh, uh, light response is restored, and these retinal ganglion cells can start to fire when the light is shined to the retina. Another beautiful example for retinal restoration comes from Raphael and colleagues, where they perform photopharmacological control on neural activity by using third generation photo switches such as diethylamine or ozodiethylamine. And what they do in these experiments is they take a triple knockout mouse that has mutations for rhodopsin, melanopsin, and CNGA3. And once they target this photosensitive molecule, photoswitching molecule to the bipolar cells, they can restore light responses for blue and white light in these animal models, as evident again by the raster plots. We also have beautiful examples for cellular transplantation therapies with the retina and then using MEA, where, for instance, Santos Ferreira and all what they have performed is they have taken CPFL mice where they have a photoreceptor function loss, and they then harvest healthy broad cone photoreceptors. They transfect these animals with these photoreceptors. These photoreceptors integrate, and they are able to show light response on MEA experiments. Uh, Other experiments, for instance, from Ligali et al., they use the approach of channel rhodopsin, again in RD1 mice. In this case, they electroporate the channel rhodopsin into bipolar cells, and then again, by shining the blue light onto these retina, they're able to restore the light response properties. Now, of course, you can also use retina not just for restoration purposes, but you can also try to understand how the neuronal circuit is connected. And to do this, for instance, publications by Field and Freeman are very beautiful examples where they identify the location, type, and strength of the functional input of each cone photoreceptor to each ganglion cells using MEA technology. Another aspect that we can study using MEA is the aspect of visual development. For instance, the example of Elstrode et al., they showed that certain aspects of vision, like direction selectivity, it develops even before eye opening and develops independently whether there's light present or not. Now, to highlight more the MaxWell MEA, I will focus more on a disease phenotype study using our system, and I will focus on FRMD7. So in this publication by Yorihara et al., the authors investigate whether FRMD7 is the key regulator establishing optomotor response in human and in mice. So what we know is that mutations in FRMD7 cause the human disease of X-linked infantile nystagmus, and this disease represents by abnormal eye movements and selective loss in horizontal direction selectivity. Now, as we can see in panel A, when we look at mice, control mice, these show nice horizontal eye movements and vertical eye movements, while in the FRMD7 transmutant mice, horizontal eye movements are gone, whilst the vertical movements are still maintained. And this is also evident in the video here on the right. Now, the same gene mutation in humans, again, in the horizontal eye movements are lost in FRMD7, but the vertical eye movements are still present. Now, in order to perform these experiments, the scientists had the question, is this phenotype already evident at the level of the retina? And for that, they used the MaxWell hardware, where they used the tissue holder, as Marie mentioned previously, and an upright microscope coupled to a pico projector for light stimulation. Then they have dissected the retina of these wild type and these mutant animals. They have placed it on the MEA, and they have determined the response properties of on-off direction selective cells and on-direction selective cells. We do know from the literature that the slowly tuned on-direction selective cells are the major drivers of the optomotor and the optokinetic reflex. And as evident by the polar plots, horizontal direction selectivity is missing in the mutant animals, both in the on-off direction selective cells and the on-direction selective cells. And this is also evident if you look at the raster plots, that there are no ganglion cells with responsive properties towards horizontal moving bars. Now, an interesting fact is that this lack of horizontal direction selectivity is already present at eye opening. And also, when scientists look at actually the morphology of these cells, they see that, for instance, the hemiprine cells, the starboard hemiprine cells, the major drivers of these direction selectivity has normal morphology, and they don't show any abnormal processes. Now, when scientists try to perform and to cross-validate these results using patch clamp, they used a genetically labeled mouse line, the HOX-D10, and in this mouse line, on-direction selective and on-off direction selective cells tuned towards forward motion but labeled by GFP. They patch these cells, and what the scientists have found that in on-off control animals, you have direction selectivity in forward motion and horizontal motion, but in the mutant, this direction selectivity is lacking. And the same results can be seen for the on control and the on DS cells for the F1D7 mutant animals. Now, one can ask the question that is this really a phenomena that is causing the disease in humans? Well, what we know is that symmetric inhibitory connections established during the first postnatal week is reorganized into an asymmetric inhibitory connection during the second postnatal week in mice. But now we also know that in the F1D7, this does not happen. And the other question is F1D7 dysfunction in starburst cells causative of the loss of horizontal optokinetic reflex in humans? And one can argue that based on the results of the papers, yes, but also one can argue that because the neuronal pathways controlling the optokinetic reflex is highly conserved among species, in non-human primates, F1D7, as in mice, is expressed in CHED-positive retinal ganglion cells, and that the vertical optokinetic reflex is still present in human patients also, as in mice. Now, with this, I would like to conclude. So in conclusion, by using the MaxWell MEA system, you can study the retina in context of visual restoration, development, visual processing, disease, and now it is also possible to study with this system retinal organoids. And that I would like to thank you for your attention.
Thank you very much, Szilard, for a very nice presentation. So, it was very good to see how high-density microelectrode arrays can be used for retinal studies. And also, the study that Szilard mentioned has a validation of high-density microelectrode array results with patch clamp results. So, this is really, really nice. Okay, so now let's go on to our next section, which is a question and answer. Thank you so much for those who sent us messages via the chat.
Thank you very much for the questions.
Yes. Okay. So, first question that we have here is yeah, so it's about how to use Max One system for retina experiments, meaning how do you prepare the sample and do the experiments themselves?
So the experiments themselves you do in the following way. So first of all, you would have your model organism of choice, either mice, rats, salamanders, or fish, depending on what you're studying. You would inoculate the animal, you would dissect the retina, of course, and all of this has to be done in dark, in full darkness, or with dim red illumination, so you don't have photo bleaching. Then it is very crucial to clean the retinal surface properly. What I mean by this is to remove the inner limiting membrane, the vitreous, so the contact patch between the retina and the MEA has a clean surface. Then you place the retina on the MEA, you place it in the recording chamber, you start your perfusion, and your perfusion fluid aims or ringer has to be set at 35 degrees. And then, of course, after some time adaptation, you perform your experiments using an upright microscope and some sort of light projection device, either a pico projector, a monitor, or whatever is available in your laboratory.
Cool. So basically, because the retina is curved, and you use kind of the tissue holder to flatten it onto the microelectrode array, and then hold it there for several hours or even more than a day to be able to get these activities from retinal ganglion cells. And you also mentioned about light stimulation. Can you elaborate more on this? Meaning, can we actually um, project different types of patterns, and do these have value in determining different types of retinal ganglion cells?
Yes, of course. So by selecting your stimuli of choice, you can distinguish, as I mentioned, direction selective cells from non-direction selective cells, and these usually you do with bars moving across the receptive field with potentially different type with different speeds in order to distinguish like fast tuned cells from slowly tuned cells. You can have light flashes in order to distinguish between on cells, off cells, on transient cells, on sustained cells, and so on. Then you can project little marching squares or white noise in order to establish the receptive fields of individual retinal ganglion cells. And now you can also, for instance, have different types of natural environment stimuli to try to distinguish your cell types. So it really depends on the question that you want to ask and to what depth you want to analyze your cell types.
Cool. We have a kind of a related question here to stimulating retinal ganglion cells. It says, can we use electrical stimulation function to study the retina without using light stimulation?
Yes, so the Maxwell chip also allows you to stimulate in 32 with 32 electrodes at once, and this is very easily done. You just place your retina, as I told you before, and instead of using light stimulation, you just configure the electrodes where you want to electrically stimulate, and you look at the readouts for after electrical stimulation. So that's easily doable on the retina.
Yes, and maybe we can elaborate more on the protocol and how to do this later by email. I will send an email later. Okay, another question related to light stimulation is does light stimulation induce noise or artifact in the electrodes?
So our chips are designed to have a very low noise. And as long as the objective that you use on the microscope focuses the light path only on the recording surface and not the adjacent surface where you have the amplifiers, you have very little to no noise on normal photopic levels for retinal light stimulation.
Yes, maybe to add on to this, you can also use optogenetics with our microelectrode array Max 1 and Max 2. So that is an indication that light stimulation is compatible with the system. Okay, another question here. How does the retina holder work? Wouldn't it prevent oxygenation during the holding itself?
So on the retina holder, you have a dialysis membrane, or yes, you glue on a dialysis membrane, and then this dialysis membrane, of course, when the retina holder is touching the retina, allows for nutrients to still flow to the retina.
Yeah. And for surface coating, do we need polyl lysine or equivalent?
For retina application, you don't need any type of coating. You just need to have a clean surface, meaning that you clean the chips between use, and that's about it.
Yeah, and the tissue holder anyway keeps the retinal ganglion cells really flat on the
Yes.
electrodes. So yeah. Okay. Yes. So here we would like to use photoacoustic stimulation to stimulate retinal ganglion cells. Are there any studies that you know of that has been done using this approach?
Sorry, photoacoustic.
Photoacoustic stimulation.
Not as far as I know of these studies. They might be, but I personally, sorry, I don't know about these studies.
Yeah. So probably we will get back to you later by email. Okay, and I think we have our last question here. There has been already multiple works using microelectrode arrays for retinal studies. What is the advantage of Max One for doing retinal research?
The advantage of Max One is truly the high density because we have center to center from two electrodes. We only have 17.5 microns. So the real advantages come when you're trying to look at the receptive field of the cells. So meaning you can much clearly define these. And also the other advantage is that when you perform a spike sorting on the retina data, which you have to do, it's much easier to separate clusters because you do see the signal from one cell of multiple electrodes. So then you can have much clearer data.
Okay, that's it. Thank you very much to everyone who asked questions. We will answer your question for the rest we have not answered today by email. And stay tuned for our next webinar. You can contact us directly here in this email address info@mxwbio.com. You can schedule a call. You can even reach Szilard directly by going into our schedule here, calendly.com/mxw/call. And you can also visit our website for more information, www.mxwbio.com. Okay, so thank you once again, everyone, for attending our webinar. I hope you learned a lot from how our systems can be used for retina research. And I would like to thank as well Dr. Szilard Sajgo for a very nice presentation today.
Thank you very much.
Yeah, thank you, and see you next time.
Bye bye.
Bye.